recording, right? Alright, so for this question, we are looking at, is it a monohybrid or a dihybrid cross? Dihybrid, how do you know? Two characteristics coded for by two different genes. Okay, so you have first one, gene R, right? And it is responsible for what sort of characteristic? Okay, leaf shape. Alright, now the second gene is gene T, responsible for plant height. Okay, now over here, what is the point? Oh yeah, let's analyze the question first. So you have two parents, right? One is a round leaf tall plant and the second is a round leaf dwarf plant. Based on this, what can you do about it? They give you the phenotype, what can you do about it? Right. You try and deduce the genotype, so you use blanks if you're not sure, okay? So over here, I know that the round tall plant. So round means big R, and then a blank, and then tall means big T, and then a blank. Now then after this, round, again, big R, blank, and dwarf, small t, right? So this is what you know so far. And after that, they gave you the offspring ratio. So this parent that's unknown, cross with this parent that's unknown, I get these offspring. Okay, what is the point of giving them offspring numbers? You try and figure out the ratio. So based on this, what is the ratio? Okay, so you realize that the ratio that you get for this dihybrid cross is 3311. Now, do you know of a dihybrid Mendelian ratio of 3311? Not really, right? So what do you do about it since you don't know? Very good. So what you can do is you can, instead of considering these two gene locus together, that means the two characteristics together, you can separate the gene locus, right? So you focus on the first gene locus, then after that the second. So you treat it like as if it's a monohybrid question. Alright, so if we look at the gene locus for leaf shape, right, what is the ratio that you get in the offspring? So let's take a look at the leaf shape first. You take the round leaf, and then you take the oval leaf, okay, you group them together. So, so the first gene locus, right, so the gene for leaf shape, right, oval is to round. If you add them together, right, you get about 79 is to 245. So the ratio is approximately 1 is to 3. Something like this, or 3 is to 1, whatever. So if you, is this a ratio that you're familiar with? 1 is to 3, 3 is to 1? Yes. So what is the cross likely to be, the parents? Okay, it has to be, the parents has to be two heterozygotes that are crossed together in order to get an offspring ratio of 3 to 1. Okay, so you can go back up to the unknown and you can fill in that blank. Okay, so the blank has to be, I mean, don't tell me RR lah. Big R or small r? Small r, small r. Okay, so that's how you do it. Then we talk about the second gene locus, right? Now let's consider the second gene locus, which is the gene for plant height. Right, so the gene for plant height, let's do a calculation based on the numbers. So you have tall plants and then you have dwarf plants. Okay, so I do a calculation. What is the ratio that I get? Wait, let me just write it out first. So tall is to dwarf. It is approximately 163 is to 161. Okay, so for genetics, you have to bring your calculator, okay, for exams. And it is approximately 1 is to 1. Now, based on this Mendelian ratio, what is the parents that are being crossed to get a 1 is to 1 ratio?
Heterozygous and homozygous recessive.
Very good. It has to be a heterozygous individual with a homozygous recessive parents. Okay, so over here, these are the parents that we are talking about. Okay, so now that you know the parent is a heterozygous individual at this gene locus and a homozygous recessive, you go back to the top, right, and you fill in the blanks. So it should be a small t here and you already know it's two small t's here. Okay, so based on this, you can get the answer already. The answer is A. Alright, so what is this technique that you are doing here? Can you write? Okay, this sometimes for genetics, what you need to do is you separate the gene loci. Okay, or in other words, you consider each gene locus separately. Okay, instead of considering them together. Alright, so you do this when the ratio is not familiar to you. Can? Alright, any questions? If not, then I'm gonna move on to question number 6. Alright. Oh, absentees. Can somebody take photos? I mean, I hope they watch the recording, but in case they don't, someone take photos and send to the group chat. Okay, okay. Alright, so moving on. So she's in the same grade. Alright, now this question number 6, I think some of you have problems with this question. So anyway, over here, this is a monohybrid or dihybrid question. Dihybrid, how do you know? Two characteristics, right? So over here, the first characteristic is for seed color. The second characteristic is for seed texture. Okay? Now, basically they give you 4 different crosses. Okay, so you have 4 different pair of parents, and then their respective offsprings. Okay, is tabulated. Okay, so what can you do about this? What can you do about this? They give you the offspring, so what can you do about it? Okay, you can try and figure out a ratio and see if it's a ratio that's familiar to you. Okay, so the first one is about 1, 1, 1, 1. Okay, 1, 1, 1, 1. Right, something that you're familiar with. The second one is 3 here and 1 here, but the rest is 0. Right, the third one is about 3, 3, 1, 1. Okay, and the last one is basically all of the yellow and brown. Okay, so you try and figure out a ratio, but unfortunately, I think you're only familiar with the first and maybe the third. Okay, and of course all lah. Okay, maybe the last one is the most obvious. Okay, I would say the second one is second, third, maybe a bit more tricky. Now the first question is very important. Which alleles are dominant, and explain your answer. So based on the analyzing the results of the crosses, you should be able to tell which alleles are dominant. So let's take a look at the first gene locus between the allele for green seed and the allele for yellow seed color. Which one do you think is dominant?
Yellow and green, brown.
Yellow. It should be yellow, right? Okay. The second one in seed texture is the allele for round dominant or the allele for wrinkled dominant? Round. How do you know? When they say explain your answer, what do you think you need to do? Okay, yes, you have to cite data, basically. So you have to cite relevant crosses to support your answer. Okay, of course you can cite 1 cross. You can cite 2 crosses, you can cite 3 crosses, up to you. But actually cite 1 cross enough already. If you look at cross 1, 2, 3, 4, which is the best and easiest cross to cite? 4. Right? Why would you choose 4? Some of you may choose others, okay, but 4 is really the easiest, most straightforward. Why 4? Right, you look at the parents, you see the parents, they look totally different from each other. Right, but when you get the offspring, you only get yellow and you only get round. So what happened to the greenness and what happened to the wrinkledness is gone. So from there you can give an explanation as to why have they disappeared. It's because they has to be recessive. Okay, so what can you write? Let me see. Okay, so this is what you can write. So 2 marks. 1 mark is the state. The second mark is the explain. Okay, so cross 4 is the best site. Okay, so the key idea that you should talk about for cross 4 is that you only get this result. Okay, that means all yellow and brown. You do not have any that's green and wrinkled, green or wrinkled. So because of that, the LUs for the green and wrinkled has to be recessive. That's the main idea. Any questions? Okay, I think maybe small handful of students will choose cross 2 and 4 to explain. I don't know. I think some of you for cross 2, I think some students will choose cross 2 because yellow and yellow parents and suddenly there's an appearance of green. Okay, so it has to be a certain genotype for the parents to get green seeds, right? So you can talk about that, but I think that means only for one gene locus. Okay, so if you choose cross 2, you can only settle the answer for one gene locus. Whereas if you choose cross 4, it is better. You can talk about both gene locus together. Alright, so anyway, Ken? Are you okay? Okay. Alright, now let's continue. So the next part of the question is using suitable symbols, state the genotype of the parents. Okay, so I saw that some of you were a bit struggling with this. Okay, so what do you need to do? First of all, let's assign alphabets, okay, for the because we are supposed to use suitable symbols. The symbols that you use may be different from mine. It doesn't matter, okay, as long as you have a key somewhere. Alright, so in terms of this question, since the dominant allele, okay, the dominant allele is yellow, so I'm gonna put it as big Y, okay, for yellow. And then for green, I'm gonna use small y. Alright? Now seed texture, brown is dominant, I'm gonna use big R. Okay, wrinkled, I'm gonna use small r. If yours is different, never mind. Okay, just make sure you have a key somewhere. Alright, so based on this, alright, I am going to take a look at the parents. Okay, what is the genotype of the parents? So using the same method as before, I'm going to use some blanks, okay, to write out what is the possible genotype of the parents. So the first one, yellow and brown. Okay, can you tell me what is the possible genotype? You can say blank if you don't know. Big Y and blank. Big R, blank. Green wrinkle.
All are recessive.
All small, R, Y, Y, R, R. By the way, your Y must look a bit different. Okay, otherwise the marker cannot see capital or small letter. Okay, so how do you know what is the unknown parent, the yellow and brown?
The ratio.
You look at the ratio, very good. So if you look at the ratio, what is the ratio?
1:1.
1:1. So what are you doing here? What you're doing is you again, okay, what you can do is you separate the gene locus. Okay, or if you know the ratio 1:1:1:1, it is a heterozygote at both gene locus, right? So if you get a heterozygote at both gene locus, right, and you cross with a homorecessive, you should get a 1:1:1:1 ratio. So this is a known Mendelian ratio. Now for the second one is the most difficult. I'm going to skip the second one first. Okay, so I'm going to talk about the third one. Now for the third one, it is a ratio that is 3:3:1:1. It's something that you don't know, but nevertheless, let's figure out the parents first. So the parents is yellow and brown. So again, I'm just going to put this. And the other parent is yellow and wrinkled. Okay, so far you got this, right? So how do you figure out what is the dash? What's the blank?
Ratio.
The ratio 3:3:1:1. I don't know. I don't know what's 3:3 and 1:1. So what's that? You need to separate the gene locus. Okay, so you need to separate the gene locus. So this is what I'm gonna do. Okay, I'm gonna talk about offspring phenotypic ratio. Okay, so I'm gonna have another column for that. So right now I'm doing cross tree. Okay, for the offspring ratio for cross tree, let's talk about the first gene locus. That means the color. Yellow is to green. Is how? What's the ratio? 3:1. Right. If you do a calculation, is 470 is to 161. Approximately 3:1. Then next one, you look at round is to wrinkle. The ratio is round to wrinkle 1:1. It is about 298 is to 333, which is approximately 1:1. Right? So based on that, okay, we talk about the first gene locus first, which is for color. How do you get a 3:1 ratio?
Both are heterozygous.
They have to be both heterozygous. How do you get a 1:1 ratio for the other gene locus?
One heterozygous and
One heterozygous, the other recessive, homorecessive. Okay, so that's how you do it. Okay, so again, separate gene locus. Now we do the cross number 4. Cross 4, it is green. So what is the genotype if it's green? Recessive. So recessive. And then the round is?
both have to be dominant.
Both have to be dominant. Yes. Okay, wait. We don't know yet, so we just put this first. Alright, and for the yellow wrinkle,
Big Y.
big Y, okay, dash, and then small r, small r. Right, so this is what we know from just looking at the parental phenotype. But then later on, we need to see the results, okay? To get results of all yellow and all round.
Must all be heterozygous.
Okay, it must all be homozygous. Okay, so based on the offspring ratio, it has to be all homozygous. Alright, so you need to look at the offspring also. Now let's go back to cross number 2. Okay, I think this is the most difficult. So we look at yellow and round first, the first parent for cross 2. Yellow round, I know it is big Y dash, big R dash. The other yellow and round is also big Y dash, big R dash. So that's what I know from the parent so far. Of course, the ratio that is in cross 2, let me see, is 3:1. So what I can do is I can separate the gene locus. So if I separate the gene locus, yellow is to green is the first gene locus. It is a ratio of 3:1. Okay, the other one, round is to wrinkle.
0. Wait.
Basically, it's all round, huh? Is it all round?
Yeah, it's around.
Yeah, so it's all round. So we talk about the first gene locus first, right? To be giving a ratio of 3:1 in the offspring, the parents have to be heterozygous at both gene locus, right? And how then do you get all round for the second gene locus?
Both are heterozygous. Wait a
Yes.
second.
Two.
No.
It can be R,
Yeah.
R, or R, or
Yeah.
R.
Yeah.
All big R or one
Not
of them
not
is as well.
heterozygous.
Exactly. It can be both big R's, okay, and giving you all round, or it can be one big R and one small r. So there are two possibilities, okay? So the first possibility is like this. The second possibility is this one or this one. Do you need to give both answers? Like, do you need to give both answers? Yeah. Yes, you have to give both answers. Yes. Okay, so make sure you have both answers. Can or not? Is it doable? So genetics not so bad, right?
It's fun.
It's fun, but you need to do a lot of practice. If you don't practice, you seriously will die. So better practice. Okay, Ken? Anybody still needs the screen? Can you take a photo? Alright, moving on. Now, the next question is question number 7. Alright, so basically you have two heterozygous pea plants, right, grown from round yellow seeds, and the plants were crossed. So this is the numbers of the offspring. Does it look familiar?
933.
Yes, it's actually a 9331 ratio. Okay? Okay, so basically, if you look on further, you know that there are two gene locus, right? The first gene locus is for seed shape, which they use alphabets big A, small a. And the second gene locus is for seed color, that they use big B and small b. Right? So based on this, right, you can figure out what is the parent's genotype already. What is the parent's genotype? They are heterozygous, so big A, small a, big B, small b. So it is two double heterozygotes crossed together. Okay? Then you get a 9331 ratio. Is this Mendelian ratio? Yes. Okay? Now, the first question, I don't know whether you understand what is this question, but it's commonly asked for Cambridge. Okay, you see it's a Cambridge question. First question is, they say the expected phenotypic ratio for this cross is 9331, right? Why is it that the ratio is not exactly 9331? If you look at the offspring. Okay, so we call this, there's a name for this. This is called the, if you do chi-square test, this is called the expected ratio. Over here, what do you call this? What do you call this? This is called the observed numbers. Can you see? Alright, so why is it that the expected and the observed is not exactly? If you want the expected to be exactly, this one should be 270 and then 90, 90, 30. Can you see it is not really exactly 9331? How come it is not exact?
Fertilization rate.
Exactly. So, okay, let me give you another example that's easier to understand, okay? So if you have a coin, you flip the coin, heads and tails. Okay, let's say I flip the coin 10 times. Right? What do you expect to get for heads? You expect 5 times you get heads, another 5 times you get tails. That's the expectation, right? But in reality, do you get 5 and 5? No. Okay, you may get, example, this is expected. Maybe the observed, you get something like with 7 and 3. Does it mean that the coin got problem? Is it a biased coin? No, may not be. Okay, why? It's just by chance. So this is a chance event. Okay, whether you get head or tail. Now, how do you make it closer to the expected ratio? Very good. You widen the sample size. So instead of flipping the coin 10 times, maybe I flip it 100 times. Then you will realize that it will get closer to the expected if it is a fair coin. Okay, so this is the same idea. So what can you say for these two reasons? Can you put a star? It's a Cambridge question, commonly asked. So the first reason you can say due to chance event. Okay, example, you can talk about random fertilization. Right, if you don't understand random fertilization, let me give you a figure here. Here. So previously you did something like this, right? You cannot guarantee that you're gonna have this gamete from this parent and this gamete from this parent fusing together. So it is by chance that you have this gamete form and then this gamete form and then they fuse together. You get what I mean? So this is what we call random fertilization. Alright, so now what is another reason? Okay, good. So talking about sample size. The sample size needs to be larger to get closer to the expected ratio. Okay, so you need to have a larger sample size. Any other possibilities? Very good. Environmental effects. So it could be adverse environmental conditions. So when you're doing the experiment, sway, right, some seeds may have died. By the way, did I tell you that you guys were very lucky that your flies, your prep was on Tuesday, right? I did a prep with 302 on the next day, Wednesday. So many flies died in just one day. You know why? Now we realized why already. Because it's too hot. It was too hot. So we shifted them to a cooler place, and the next day we came back, the flies are still doing well. You know, from your experiment that day until 302's prep, wow, so much difference in the flies. They were moving so slowly, right? And those who were like a lot of flies didn't wake up. I think it's too hot. Yeah, so they are very weak. I don't know whether the experiment will be successful, but anyway, we shifted them to a cooler place and we realized that a lot of flies are still doing okay. So it's really temperature effect. So environment can affect your living organisms if you're doing genetics experiment. Okay? Anyway, there is a last one that is quite lame, I think, but Cambridge accepted it. Okay, it's called incorrect counting. Okay, but of course I think that Cambridge accepted because it is possible. Right now you're not counting 5 times heads, 5 times tails, you're counting so many. Right? So it could be a human error or something. Okay, so anyway it's accepted. Any other points you want to ask? Is it okay? Can Now the next one, I think I expect you to know how to do already. It's a genetic diagram. Can you just check your answer? Okay, now can you please make sure that you write down where's the marks allocation. So 1 mark is for genotype of parents, right? They gave you the phenotype, you have to write the genotype yourself. 2nd mark is for gametes that are circled. Okay? You need to draw 4 gametes because of independent assortment and segregation. Okay, so you get 4 possible gametes from this genotype. The next one, next mark is 3rd mark. The Punnett square, which includes the random fertilization, right? To get the offspring genotype and phenotype. And the last one, 4th mark, phenotypic ratio. Okay, so genetic diagram usually 4 marks. If you see a 6 mark question,
It is like a few generations.
yeah, perhaps you have to draw until F2 generation. Okay, that's a possibility, okay? Yes?
This question is a bit unrelated, but how come if like round and yellow seeds are like the most common, how come all the peas in the supermarket are all green?
Oh, I think they deliberately, maybe different, what you call it, different breed, different species they are looking at. Or sometimes they also artificially breed and cross to create some phenotype that looks nicer. Yeah, especially when it comes to breeding dogs and everything. Yeah? Is there a typo? What? Oh, there's a cross here, yes. And is there a typo? Where? 3rd row, 4th column. Oh. So it should be small b. Is it? Oh, wrinkled yellow. Any other typos? Okay, so anyone taking photo? Can? Is there any problems with the cross? Any question? I mean the gametes, yes. If you want, you can draw a cross here. Somebody asked, do you need to draw gender, the symbol here? For this case, yeah, they never say male, female, so not necessary. But later on, you'll see questions that they actually tell you, this is the female, this is the male. Then you have to talk about the symbols. It's easy to remember, right? The symbol, it kind of makes sense to me. Yeah? Scroll up, okay? Okay, then the last question for today, and then you'll have the rest covered during your lecture later. Can I move down now? Anybody still needs? Okay, so the next question here. Right, in another experiment, and this time you have a different plant type, it's called the bean plant, right? And it is two heterozygous plants with brown and yellow seeds were crossed, okay? And so that's all that you know. Of course, you get a ratio, and this ratio, right? What do you expect when you cross two heterozygote plants? You're supposed to get 9331, right? But how come so strange one? When you cross these bean plants heterozygous, right? You don't get 9331. Okay, so what do you notice about this offspring phenotype? There's another color that appeared, right? So if you look at the round, there are two gene locus, okay? So please split it up. If you're looking at the round versus wrinkles gene locus, is it same or is it different from the pea plant? It's the same, right? What can you do about it? What you can do with these numbers is you separate it into two gene locus and you see the ratio, right? So if we look at the ratio for round to wrinkle, let's do that first. So for the round is to wrinkle, can you do a calculation? What is the ratio?
3 to 1.
It is about 3009 is to 991, which is 3 is to 1. Right, so how do you get a 3 is to 1 ratio? It has to be a cross between two heterozygote parents. Okay, now the next one, you look at the gene locus for, what you call that? Seed color. Alright, you have green now, and you have yellow, and you have white. Which is very strange, okay, because there's an appearance of a new color which is green. Okay, and if you calculate the ratio, it is about 991 is to 2003 is to 1006. It is approximately 1 is to 2 is to 1. Okay, now based on this ratio 1 to 1, does it look familiar to you?
Yes.
It's a non-Mendelian ratio, okay, it's 1 to 1. Now where do you see 1 to 1 before? Okay, incomplete. Is that all? You see it in incomplete dominance, you also see it in codominance. Right, now what is the difference between incomplete and codominance? This one, a lot of students ask me all the time, okay, codominance versus incomplete dominance. What's the difference? Codominance means? Yes, alleles are equally expressed in a heterozygote. Right? Example of codominance, the most famous example, your blood type IA, IB, alleles, they are heterozygote. In a heterozygote, alright, your blood will have both antigen A and antigen B because they are equally expressed. Right? So that's codominance. Another example in your notes is the coat color of the cattle, I think. So if you have CR allele for red coat color in the cattle, CW allele for white coat color in the cattle, if it is a heterozygote, what do you think the coat color of the cattle will look like?
Spots, red and yellow.
Yes? Huh? Yes, that word. Okay, never mind. You keep that thought first. I don't want to tell you yet. For incomplete dominance, what is the difference? This one is equally expressed. How about this one?
This one, for example, you dominate and you might green, but you get pale green.
Something like that. So what is the key word? The phenotype is?
Partially expressed.
The phenotype is intermediate. The word that I'm looking out for is it is intermediate. Okay, the phenotype of a heterozygote, okay, is intermediate of its homozygous parents. Of homozygotes. Okay, so example in your notes is the flower color. Right, CR allele is for red color flower, CW allele is for white color flower. If you get a heterozygote CR, CW, okay, now look at the difference here versus here, codominance versus incomplete. For codominance, CR and CW heterozygotes, what is the phenotype of the coat for the cattle?
Red and white.
Yes, red and white. So it's basically patchy lah. Red and white. Right? For CR and CW heterozygote for incomplete,
Pink.
you get pink because it's an intermediate. So in this case, this question, is it incomplete or is it co?
It's incomplete.
Incomplete. Who is the heterozygote?
Oh, the yellow is
The yellow. It has to be the yellow. Okay, so this is a heterozygote. Okay, it is CRC. Okay, depends on what alleles you want to assign. Let me just anyhowly assign. Okay, maybe this one I put CG for the greenness. Okay, CW. So this one CG. Okay, so this is my anyhowly assigned alleles. So basically what I want to point out is a lot of times in the question when you see codominant incomplete, usually they will put like a C, like some alphabet and it becomes a superscript. You know what I mean? Yes, something like that. Okay, so last part. Before, don't need to clean because you're staying here, right? Okay, last part before we end. How is the inheritance of the characteristics of shape and color of seeds in the bean different from that in the pea? Okay, so 4 marks. How do you separate the marks? How do you separate the marks? 4 marks. Guys, you know this. How to separate 4 marks based on this question?
different characteristics.
Different characteristic, good. So the seed shape will be 2 marks. Alright, so 1 mark, 2 mark, and then the seed color, okay, will be 2 marks. Alright, I'm going to show you the answer later. Can we just discuss what is the outline of the answer? So for the seed shape, what do you think is expected in your answer when they say how is the inheritance of the characteristic different? So for seed shape, it's not different lah. So we can talk about seed shape quite easily. So what do you think is expected in your answer? Ratio, very good, because there's a reason why they give you data, right? You have to tell me the ratio. Right, you need to tell me that what is observed in both peas and beans. Okay, the ratio is 3 round is to 1 wrinkle. Okay, which means that the parents have to be crossed between 2 heterozygote parents. Now that's 1 mark, which I think a lot of people will get that 1 mark. Okay, anything else that you think you need to talk about? Why do you get 3:1? Because the alleles, okay, show complete dominance. So the allele for the what? Round seed is completely dominant over, okay, the allele for the wrinkled seed. So what is the keyword? The keyword is complete dominance. Okay? This one shouldn't be a problem. The one that is a bit harder is the next characteristic. So lastly, the seed color characteristic. Again, what should I start with? Ratio. Good. So for the ratio, I'm gonna say that for the P, okay, I get 3 yellow is to 1 green. The ratio is P, 3 yellow is to 1 green, because I cross 2 heterozygotes together. Okay, versus in beans, I get a ratio of 1 green is to 2 yellow is to 1 white. Okay, and what do you think is crossed? Okay, they already tell you 2 heterozygotes anyway. Okay, so again, the first one. Okay, so both is crossing 2 heterozygotes, but one you get 3:1, the other one you get 1:1. So the reason for that is the allele relationship. So in your pea plants, basically, the allele for yellow, okay, is completely dominant over allele for green color. Right, versus in the bean plants, right, the allele for Which is the dominant, so-called the more dominant allele? Which color? Is it green, is it yellow, or is it white? Green. Right? So the green is incompletely dominant over the white, so you get yellow. Okay, so the allele for the green is basically incompletely dominant over the allele for the white. So last marking point, therefore the beans that are heterozygous, okay, will be yellow. Alright, so this is basically the outline. If you want to see it in words, I can show you and then you can take a photo. Anyway, it's the same lah. Alright, Ken? I hope you all didn't think this is epistasis. Some students will write it's epistasis. Okay, I'm gonna show you and then yeah, you can go for a break. Okay, so that's all for today. So after that lecture, we cover 8 and 9. So